Welcome back. We're going to pick up where we left off with lecture 1B, where we looked at the kinds of chemical bonds. And in this lecture, we're going to take a look at chemical reactions, the different types of chemical reactions, and a little bit about how enzyme catalysts can speed up enzyme reactions. A chemical reaction occurs when new bonds form or when bonds are broken. And chemical reactions are made up of reactants, which you will find on the left-hand side of the arrow. So in this figure, our reactants are 2 hydrogen molecules and 1 oxygen molecule. They're going to interact with each other. A chemical reaction is going to take place. And then you're going to have on the right-hand side of the arrow, the products. And in this case, in this reaction, our products are 2 molecules of H2O. We're going to break these bonds that exist between the hydrogen molecules. We're going to break these bonds which exist between the oxygen atoms. And then we are going to, through the chemical reaction, reform brand new bonds and create products. There are numerous types of chemical reactions that have been classified in chemistry. I want to point out the 3 most common types that will occur in the human body. These are going to be your synthesis reactions, otherwise known as anabolic reactions, your decomposition reactions, also called catabolic reactions, and then your exchange reactions. In your synthesis reactions, you've got your reactants A and B. They're going to interact with one another, and a bond is going to form, and you're going to produce the product AB. Decomposition is the opposite. You will take the reactant, which is AB, and you're going to break the bond between them, and you're going to break them down into their constituent components, in this case, A and B. And then in the exchange reaction, we have AB and CD interacting with each other to form brand new products, which are called AD and BC. Now, the way I've written these letters in the exchange reaction is very intentional. It's not a random thing. In the exchange reaction, you are switching or you are exchanging B and D. They are kind of swapping places, and they are forming brand new products. So these are the 3 most common types of chemical reactions that I want you to learn for your exam. These 3 types of reactions go on constantly in the human body, and the combination of all chemical reactions that occur in the human body is referred to as metabolism. I really like this figure because even though it doesn't deal with exchange reactions, it does illustrate a fundamental concept that you need to learn as a physiology student, is that catabolic reactions drive anabolic reactions. Catabolic reactions are going to break larger molecules down into smaller ones, and they are going to generate energy as a result of that. That energy is usually in the form of ATP. And that energy that we make through catabolic reactions is used to take smaller molecules and add them together to form bigger ones. So that ATP, that energy, is used to drive anabolic reactions. Both anabolic and catabolic reactions are going to produce unusable energy in the form of what we call heat. This heat is released into the system, and it's what produces our internal body temperature. So metabolic reactions creates our internal body temperature. And then finally, you'll also see that catabolic reactions will generate what are referred to as wastes, which are, again, products that the body cannot use and will try to eliminate in some form. Even though we won't do it in this course very much, when you write out a chemical reaction, you have to write them in the balanced form. You'll also notice that when reactions are shown in this course, they will be written in balanced form. And that's because we have to follow the law of conservation of mass, which I look at as sort of chemical bookkeeping. The law of conservation of mass means that the number of atoms on the left-hand side of the arrow have to equal the number of atoms on the right-hand side of the arrow. In other words, the atoms in the reactants have to equal the atoms you find in the products. And this is because you cannot create or destroy matter or energy. That is like the first law of thermodynamics, one of the fundamental laws of the universe. So again, we're not going to do a lot of chemical reactions in terms of writing them out, but if you are required to write out a chemical reaction, they have to be balanced. The reactants of a chemical reaction are continuously moving around and colliding with one another. So our atoms, our molecules, our ions, they are continuously moving and banging into each other. And this movement and collision is known as kinetic energy. Now, if the kinetic energy of a chemical reaction is big enough, in other words, if the collision between, say, 2 molecules is large enough, it can disrupt the valence electrons in those molecules, and either you'll get a new chemical bond which will form, or you'll break up a bond. This collision energy is referred to as the activation energy of the reaction. These collisions that occur in chemical reactions are critical to the progression of every single chemical reaction that occurs in our body. And the more often a collision occurs between reactants, the greater the chance you're either going to form a new bond or break a bond. I want you to think of the activation energy of a reaction as being the initial energy investment that you need to put in order to start a reaction. This is because in a chemical reaction, the reactants have to absorb enough energy in order to destabilize their chemical bonds. And making their chemical bonds unstable is necessary so that they can break new bonds, or break bonds and form new ones. This point in a reaction where the chemical bonds are unstable enough is called the transition state. And the energy required to get to that transition state, shown by this arrow here, that's your energy of activation. In some textbooks, they'll call that energy the free energy of activation, but you can call it the activation energy. It is short formed with the letters EA. Now, where does this activation energy come from? A lot of reactions get this activation energy from their environment. Like often, the activation energy is supplied in the form of thermal energy, or what we call heat. Once you get over that hump, once you invest that energy, you get to the transition state, and the bonds between the reactants are destabilized. It's very, very easy at that point for the reaction to be driven to completion. And in this case, you have brand new products made with brand new bonds. Again, as you put activation energy into a reaction, you are making the bonds in the reactants weaker. You're weakening those bonds so that your reactants enter into this transition state. Our transition state would be here, right up here. New products form as the electrons in the transition state reactions rearrange to form brand new bonds. Each reaction involves a change in energy as it proceeds to completion. This energy is actually called free energy. If the reaction liberates energy, in other words, as the reactants proceed to become products and energy is given off, this is called an exergonic reaction. And this happens because the free energy of the products is less than that of the reactants. Now, if this energy given off is in the form of heat, this exergonic reaction can also be called exothermic. All exergonic reactions are going to liberate energy. This energy doesn't necessarily have to be in the form of heat, though. It can be in the form of light. So it leads to the phrase that all exothermic reactions are exergonic, but not all exergonic reactions are exothermic. Let's take a look at the opposite. If the energy of the products ends up being greater than the reactants, you have to put energy into the system beyond that of activation energy. I know it's not shown here on this graph, but again, you have to make an initial investment of energy into every chemical reaction. If you have to keep putting energy in to keep driving that reaction to completion, that's an endergonic reaction. And that's because the free energy content of the products is greater than that of the reactants. This slide compares and contrasts endergonic and exergonic reactions. Notice again, we've got the activation energy, but the difference between these two reactions is the energy of the products. If the energy of the products is lower than that of the reactants and there's a net release of energy, you're exergonic. If that net release of energy is in the form of heat, the exergonic reaction is exothermic. In an endergonic reaction, the energy of the products is greater than that of the reactants. So you have to put even more energy in. If that energy you put in is in the form of heat, then that endergonic reaction can also be called endothermic. Two influences on activation energy and the rate of a reaction are temperature and concentration. For example, heating a reaction can increase the kinetic energy of the reactants. It actually can make achieving activation energy easier as the reactants collide faster, harder, and more often. So you can get over that initial hump
of the reaction and then continue on down to complete the reaction. I think of activation energy as that first hill of a roller coaster. Concentration can also change reaction rates. If you increase the concentration of reactants, you will increase the chance that a collision will occur more often among the reactants. And you will, again, get over that activation energy hump faster, easier, and your rate of completing the reaction will increase. So if you want to speed up the reaction rate, you can increase temperature and you can increase concentration.
A chemical reaction can get its activation energy from its environment. But this is not going to work for the human body because it's going to take far too long for a chemical reaction to acquire enough activation energy from the surrounding heat of the body. Remember, metabolic reactions are going to produce a certain amount of unusable energy in the form of heat, and that provides a source of activation energy. But that's nowhere near enough to what we need to conduct the majority of our metabolic reactions. So we need to speed up these reactions using a compound called a catalyst. A catalyst is something that lowers a reaction's activation energy, it speeds up the formation of products, and it is not consumed in that reaction. So in this figure on the right-hand side, we have two reactions. We have the top line right here in red, which is our typical chemical reaction, and the lower line right here shown in green is a reaction under the influence of a catalyst. You will notice that this activation energy arrow that I'm circling right now is significantly lower than the chemical reaction without the catalyst. In essence, you're making the hill easier to get over, aren't you? Now, you're not changing the energy of the reactants, you're not changing the energy of the products. All you're doing is changing the initial amount of energy you need to invest in order to drive the reactants to become products. And catalysts simply speed up chemical reactions. That's what we use in the human body to ensure our metabolic reactions get done quickly and efficiently. In biology, catalysts are called enzymes. Enzymes are proteins that speed up or catalyze a reaction without being consumed in that reaction. So an enzyme is a biological catalyst, and they speed up reactions that would occur naturally. Enzymes bind reactants. In this case, when dealing with an enzyme, we call the reactants substrates. They bind their substrates in an area called an active site. And achieving the transition state in the active site is so much easier when using an enzyme than achieving a transition state without one. There's numerous reasons why the active site allows achieving the transition state that much easier. We don't need to go into that in physiology. Just realize that as the substrates bind their active site, it becomes a lot easier to hit that transition state. And once you've hit that transition state, the reaction is driven to completion, the product is made, and then it is released from the enzyme. The last mini lecture in this series is lecture 1D, and it's going to talk a little bit about water, solutions, and buffers. Head on over there.